Hello, I am Professor Shrudeep De of Northeastern Hill University, Shillong, India. I shall be delivering a series of talks on electron microscopy. In my first lecture, I shall be talking about the basic principles, preparatory techniques, and some important applications of electron microscopy. In a simple way, one can say that microscope is a device through which a magnified image of an object is available. However, magnification is not the only consideration in microscopy. A far more important factor in microscopy is resolution. Resolution is the capacity of distinguishing two closely placed particles as separate entity. In light microscope, the best resolution is approximately 0.2 micrometer as opposed to 0.1 millimeter for the unaided eye. Resolution of light microscope will thus allow detection of cell organelles such as nuclei, mitochondria, etc., but not in detail. This photographic representation shows the first picture, that is A, which shows that there are existence of two particles but very undistinguished. Next one partially resolved, so two separate particles can be seen as separate entity but not complete separate entity, and in the resolved one we can see that two particles are separate entity. Resolution is limited primarily by wave nature of light or other illuminating beam. The relationship between resolution and wavelength is given by Abbe's equation. Here, the wavelength of light will determine the resolution, which is low or high. This is the photographic representation of how the resolution in a microscope is determined. Electron microscopy was developed through several electron optics research, theoretical as well as practical, and finally it was found out that wave nature of electron is there, which can be applied for development of electron microscopy by using electron as the source of illumination. And finally, Knoll and Ruska in 1932 developed the first electron microscopy. This is the historical development of electron microscope. This is a representation of the comparison between light microscopy, transmission electron microscopy, and scanning electron microscopy. In light microscopy, the source of illumination is light, whereas in transmission and scanning electron microscopy, the source of illumination is electron. Because the wavelength of electron is very low as compared to light, we get a highly resolved image in both scanning and transmission electron microscope. There is a column in the microscope which is under high vacuum. There is a gun chamber in which tungsten filament or lanthanum hexaboride filament is there, and because of thermionic emission the electrons are ejected out. The column of the electron microscope should be under high vacuum because presence of little bit of air particles in the column will disturb the passage of electrons into the sample. There are lens systems in both scanning and transmission electron microscope. The condenser lens condenses the electron beam. The objective lens makes the first image. This first image is used as an object for the intermediate lens and the projective lens, and finally a highly resolved, highly magnified image is produced. The two types of electron microscope, that is scanning and transmission, has different purpose and also works on different principles. In transmission electron microscope, electron beam has to pass through the sample, hence we need highly fine sections for studying. In case of scanning electron microscopy, the surface details are studied, so here the thickness is immaterial. This is again the image of the internal arrangement of the scanning electron microscope. The electron gun which generates electron is composed of hairpin shaped tungsten filament, one-length cylinder and the anode plate. This is the arrangement of one-length cylinder in case of scanning electron microscope. This is the way how electron beams are ejected out through the tungsten filament due to thermionic emission. The lenses seen in both scanning and transmission electron microscopy are not actually visible because they are electromagnetic field. The microscope column consists of illuminating system, electron gun and condenser lens, imaging system, image translating system. In scanning electron microscope, when the primary electron generated in the gun chamber interacts with the specimen, we have a detector which detects the secondary electron image. This secondary electron emission produces the image at point by point, line by line situation. In addition to imaging, electron microscopy has another device through which one can study the elemental composition of the specimen. This is called energy dispersive X-ray spectroscopy. When the electron interacts with the specimen surface, then several signals are coming out. One is secondary electron which gives us the image. Another is backscattered electrons which gives us image from the depth of the sample, and there is cathodoluminescence Auger electrons. The X-ray signals coming out from the sample are characteristic X-rays. When we consider the structure of an atom, we can see how outside the nucleus different shell has distribution of electrons. When electron beam falls on a specimen, then from the atom from outermost shell electrons come out to occupy the position of inner electron. As a result, some energy is lost. The loss of this energy is expressed in the form of X-ray energy. This X-ray energy is called characteristic X-rays because their energy is different in different elements. As for example, for aluminum, the X-ray energy is different from that of sodium or potassium or iron or magnesium. This is the picture showing how the electrons from inner shell is removed by the primary electron, and then the electron from outer shell, which are a high energy shell, occupies the position. The characteristic X-rays appear first as continuous X-ray, and with the help of an instrument called energy dispersive X-ray spectroscopy, the spectrum is resolved into different peaks, and these peaks can be identified into different elements by using the periodic table mode of the spectroscope. Now, after the principle of electron microscopy, we will describe about the sample preparation in case of scanning and transmission electron microscopy. In scanning electron microscopy, the samples are first prepared as 2 mm by 2 mm thickness. Then it is fixed in 2.5 to 3% glutarylate, prepared in 0.1 molar sodium cacodylate buffer. We can also use phosphate buffer in this case. The fixation may be done either through immersion fixation, that is, dipping the sample in the solution of the fixative, or it can be done in case of delicate samples through vapor fixation. In vapor fixation, a small piece of cotton is soaked in osmium tetroxide and then it is placed in a petri dish. The sample is placed nearby and it is covered and kept overnight. Thus, the vapor of osmium tetroxide fixes the sample. After fixation, the sample has to be dehydrated, and it is done through ascending grades of acetone and water mixture. That is 30%, 50%, 70% up to 100%. After the sample is dried in a drying instrument called critical point dryer. In this critical point dryer, the critical point, that is a particular temperature and pressure of a fluid, as for example carbon dioxide, is reached, and then because of the density, no density change in this critical point, the samples are dried without any surface damage. In our laboratory, we have developed another alternative method with the help of a chemical called tetramethylsilane, TMS, which can dry the samples just like critical point drying by taking into consideration the simple chemical and physical properties of the compound. TMS is inert chemical and its boiling point is almost near room temperature, that is 26 degrees centigrade, and its surface tension is also quite low. After the sample is dried, it has to be secured in a metal stub made of brass with the help of double adhesive tape or some paints. After that, the sample has to be stained or coated with gold in an instrument called ion sputter. In case of transmission electron microscopy, on the other hand, similar type of fixation, dehydration, etc. is going on, and after that the sample is passed through the infiltration medium and embedding medium combination in different ratios in an oven for 48 to 72 hours. After that, the sample is molded in a mold made of plastic. Mold can be flat mold or it can be cylindrical mold. In addition to glutaraldehyde and other fixatives, different types of fixatives can be used for different purposes in transmission electron microscopy. For fixing neutrophils, paraformaldehyde prepared in phosphate buffer gives excellent result. This is another example of sections of neutrophils fixed with paraformaldehyde in cacodylate buffer. Rabbit ciliary epithelium can be fixed with glutaraldehyde followed by osmium tetroxide. The same section fixed with osmium tetroxide alone. This micrograph shows the TS of fish muscle fibers fixed with glutaraldehyde followed by osmium tetroxide. Proper preservation of rough and smooth membranes, ribosomes, mitochondria, the osteoblast in glutaraldehyde and osmium tetroxide fixation can be seen nicely. This is the micrograph showing section of rabbit kidney fixed with potassium permanganate. This micrograph shows transverse section of fish muscle fixed with glutaraldehyde treated with hydrogen peroxide followed by osmium tetroxide and uranyl acetate. Comparison of preservation of membranes in different fixatives. Osmium tetroxide plus potassium permanganate in first picture, only potassium permanganate in second picture, osmium tetroxide plus uranyl acetate in the third picture, and osmium tetroxide only in the fourth picture. This is the transverse section of rat brain in acetone dehydration showing closely opposed plasma membrane. Because acetone is having low surface tension, hence the surface membrane is closely opposed. Same in ethanol dehydration showing adjacent plasma membrane separated by extracellular space. Thus, not only fixation, it is the dehydration which matters in case of electron microscopy. This is the photograph showing an area containing calcium has not been well impregnated with the embedding medium and has been torn out. So one has to take care about the elemental composition of the sample also while emitting it. Thus we could see that fixation can be used in different manner for different samples. Different fixative gives different result for different type of sample, and dehydration also plays a very important role because acetone has low surface tension and hence the preservation of membrane is very clear, whereas in case of alcohol dehydration the membranes are dilated apart. After the block is prepared, section is cut and it is placed in this type of units known as the grids. Grids consists of different types of meshes and is used differently in case of different samples. This micrograph shows the defects in sectioning due to bluntness of glass knife. The section is actually taken with the help of an ultramicrotome where the knife used is made of glass. Now, if the glass knife is not sharp enough and it is blunt, then this type of knife mark will come and it will disturb the structural features of the section. These are the photographs showing different types of macromolecule in human body. Now we'll talk about some important biological application of electron microscopy. Electron microscopy is used in cell biology. Three-layered structural organization of plasma membrane, inner and outer mitochondrial membrane, nuclear envelope, nuclear pore, endoplasmic reticulum, Golgi complex, etc. can be studied in detail with the help of electron microscopy. In molecular biology and nanobiotechnology also, electron microscopy finds its wide application. Ultrastructural features of soluble, fibrous and membrane-bound proteins can be studied with electron microscopy. Structural studies on nuclear envelope and nuclear pore complex can be studied. Morphology of glycoprotein and coated vesicles, structure function relationship in cilia and flagella can also be studied in detail. Bacterial appendages can be studied in detail with electron microscopy, and unstained protein crystals can be studied with the help of electron microscopy. In microbiology, classification of microorganisms, surface features of bacterial pores, surface changes induced in microorganisms by antibiotics, in botany, structural features of epidermis, algae, fungi, seeds, fruits, pollen, spores, taxonomy, wood anatomy with respect to pulp and paper industry, ecophysiology has wide application in electron microscopy. In entomology, morphology of insects, both larval and adult, taxonomy of larval as well as adult insect, developmental studies of both the types of insects, and physiology, neurobiology, etc. can be studied in great detail with the help of electron microscopy. In paleontology, illustrations of microfossils and classifications of fossils can be done with electron microscopy. In parasitology, structural features in relation to taxonomy, assessment of anti-helminthic substances and plant products can be studied with electron microscopy. Now I will show you some of my own work which finds application in some economic aspects of zoology. This is the photograph of the lateral portion of the head cuticle of the silkworm larvae Antheria asanensis. Now why we wanted to make this type of study is that we have observed that the first, second, third, fourth and fifth stages of insect feed on the hostprint leaves, but in case of first to second stages they can feed in any position, horizontal or vertical, whereas from third stage onwards the insect larvae feed on only at vertical position. So we wanted to know, is there any basis of ultrastructural features of their head to determine this? Then we have found that from third stage onwards, this type of structures are present in the head cuticle of Antheria asanensis. Now these round dome-shaped structures are called gravity receptors or dorsal campaniform sensilla, and they determine the vertical posture of the larva during feeding. This is the enlarged view of the dome-shaped gravity receptors or dorsal campaniform sensilla. When we compared with some silk moth larvae which do not feed in vertical position but they can feed only in horizontal position, we found that there is no such dorsal campaniform sensilla or gravity receptors. In case of lower stages, we find scattered distribution of dorsal campaniform sensilla, explaining the reason for their feeding at horizontal position. This is the individual picture of the dome-shaped sensilla. After observing the distribution pattern of this dorsal campaniform sensilla, we wanted to know whether it really determines the vertical posture from third stage onwards during feeding. For that, we placed a few larvae in a tray with full of horseplant leaf, and then we found that the larvae are trying to climb up and acquire in a vertical position, and their feeding rate is very low. When we put some arrangement like putting the twigs along with fresh leaves in bottle and make them to feed vertically, we found that their food consumption is really high, and all the other economic features such as the shell weight, body weight, the cocoon weight, everything improved, and their lifespan also became normal, unlike those fed on horizontal position. Electron microscopy plays a very important role in erythrocyte dynamics in metal toxicity. Here in the first picture we can see the normal shape as well as some abnormally shaped erythrocytes. In the middle picture we find the echinocyte generation, which expresses the gravity of hemolysis. We have other type of abnormal erythrocytes, that is phagocytes, then reticulocytes, etc., which will be shown in the next few pictures. This is also a kind of echinocyte, but in the early stages of development. In the second picture also we can see echinocytes, and in the third picture a number of echinocytes have been developed in rabbits with lead toxicity. This is different types of abnormal erythrocytes in rats exposed to lead. In the third picture we can see membrane internalization, which is also an indication of hemolysis. This is the ring-shaped erythrocytes. This also expresses the hemolytic nature. Now I will show some of my experiments where energy dispersive X-ray spectroscopy is used for understanding certain important things. So we all know that cornea and lens are very important for our vision, not only for us but also for other mammals. So it is surprising that no one knows in detail the total elemental setup of cornea and lens of many animals. In fact, the reason is that when someone is interested about some elements, you will go for that with the help of atomic absorption spectroscopy or other device. But if we want to know the total picture of the elements present in the cornea and lens, we have to use energy dispersive X-ray spectroscopy, and unfortunately no one has studied in this. So we have found out the total X-ray spectrum of some mammals like spotted deer cornea, spotted deer lens, gibbon cornea, gibbon lens, sambar cornea, sambar lens, hippopotamus cornea, hippopotamus lens, if we look at the energy dispersing X-ray spectra of cornea and lens of different mammals, we will find that the sulfur peak is very high. And we published these papers in biomedical letters, and we got the information that this study is important in the sense that the taking up of fine fiberless food by human being was related to the development of several corneal and lens diseases. So in that case, in our case, in Indian case, we are taking foods with fibers, and that probably for that reason the lens and corneal disease is less as compared to Western countries which take fine and refined food and do not take fibers in high quantity. Another example of our work where electron microscopy could be applied for economic entomology is UV sensitivity and coupling behavior. In case of Antheriacebensis, it is very difficult to rear them outside because the birds, hailstorm, etc. damages the insect larvae in the field. So that's why the indoor rearing becomes very much popular. But here we have seen that when we keep the insects inside the room, then auto-coupling behavior is disturbed. A ratio of auto-coupling becomes 1:5. That means one moth approaches 5 females and then only one is successful and the eggs are produced from one female. That way, the number of male or female moths are become useless. That is why the reason for low autocoupling behavior in indoor was tried. We have found that in case of wing of the moths, there is a structure which can be seen here. The first one is low magnified, second one is highly magnified. And you can see that in between the two horizontal strips, there are vertical strips also. And when we studied their refractive index and the refractive index of AR in between, and we apply the theory of optics, we find that this type of structure can act as and that this type of structure can be used for reflecting UV, and this UV is taken by the other counterpart, and as a result coupling becomes total. To confirm it experimentally, we took 100 male and 100 female outside the room, and we have observed that during twilight period, that means neither day nor night, when the UV rays are maximum in air, then the coupling becomes 100%. So we could avoid the wastage of moths in coupling. This is the photograph through UV light in the lower picture and normal light in the upper picture. In the lower picture we find the shape is not seen properly because only those area from where UV is reflected we could see some structure, whereas in the case of other, the normal light gives the detailed picture of the moth. This is the experiment which we did. In case of twilight condition, the moths coupled 100%, and in case of inside the room, the moths are scattered. They do not couple because they cannot communicate with the help of ultraviolet light. This is another example of application of electron microscopy in fish biology. This is the picture of an hill stream fish. It has been found that the water in which they are growing is acidic, but even then the fish is surviving, reproducing, and its population is not declining to a great extent. We wanted to know what is the reason, because we know that acidic pH causes a lot of harm to fishes and fish structure. This is the picture of muscle distortion in Equipinatus due to acidic pH. The first one is the normal muscle. The second one, we can see a disturbance in the muscle structure. In case of gill also, we find that in the upper picture, the acidic pH has disturbed the gill lamella. In the lower one, a normal pH, the gills are intact. This is well developed central capillary pillar cells and surface epithelial cells in D. equipinatus near neutral pH. When pH is not acidic and it is near neutral, then no disturbance is observed. This is the micrograph showing short sperm head of Equipinatus in acidic pH. Section of D. equipinatus showing the prominent length of the sperm in neutral pH, which indicates that acidic pH really disturbs even the reproductive structure of the fish. This is the micrograph of a sperm of the fish grown in near-neutral pH. That means the sperm head or the sperm tail is not at all disturbed. Now we wanted to know how the fish survives and reproduces although their structures are disturbed in acidic pH. To study this, different portion of the body was analyzed with the help of scanning electron microscopy. We find a structure which is similar to liquid crystallinity. In case of acidic pH, we don't find the complete liquid crystalline nature of the surface features of different organs, and we can find that there is a breakage. This indicates that probably liquid crystallinity is developed in case of the fish grown in acidic pH, and it is used as a molecular adaptation against the low pH. We made different studies with the help of different types of microscopy and found some evidence of liquid crystallinity development in the fish tissues. With this, I conclude my lecture. Thank you.